Good morning and welcome to this joint NGI and NBIS webinar on de novo sequencing and analysis and the service that we provide. So unfortunately, as you can see, we have one canceled talk today due to the very sad pandemic time, due to sickness. So Ignas will not present today. So my name is Michaela Asp, and I'm a project coordinator at the National Genomics Infrastructure here at SciLife Lab. So I will be your moderator during this webinar. And I also have my colleague Renuka here with me today, who is also a project coordinator at NGI, and she will operate this webinar in the background. So this webinar will be recorded and published on our YouTube channel. So as you can see in the agenda to the left, we have all our lineups, and unfortunately Ignas is cancelled. But apart from that, we have a lot of interesting talks, and we will also end this session with a Q&A session where you can ask our panel of speakers any questions you have about de novo sequencing and analysis and what we at NGI and NBIS can offer you as a service. And you also find all the approximate time slots for each speaker in the agenda, although these will be maybe a little bit skewed now due to the cancelled talk. So questions can be sent in throughout the whole webinar by using the Q&A chat. So we will answer the questions directly in the chat or at the end of the webinar during the Q&A session. So at that point, you're also welcome to ask questions by raising your hand, or if you prefer to ask it in person. So the Q&A chat and the raising your hand button can be seen at the bottom of the Zoom window. So with that, I will start this seminar by giving you a brief introduction to NGI and NBIS. So who are we at the National Genomics Infrastructure, NGI, and National Bioinformatics Infrastructure, Sweden, NBIS? So SciLife Lab hosts almost 200 research groups, 10 platforms, and 40 units. So NBIS constitutes the SciLife Lab Bioinformatics platform, and NGI is part of the Genomics platform. So NGI is a national research infrastructure and is physically located at 3 sites. So we have NGI in Stockholm, NGI in Uppsala, Uppsala Genome Center, and NGI in Uppsala, the SNP and Seq platform. So the Stockholm site and the SNP and Seq platform primarily use the Illumina technology, while the Uppsala Genome Center focus on other technologies such as PacBio and Nanopore. NBIS is a national bioinformatics infrastructure and have staffs at 6 sites, so Gothenburg, Linköping, Lund, Stockholm, Uppsala, and Umeå. And Henrik Lantz will tell you a bit more in detail on what NBIS do and the service that they provide in his talk later on. So shortly on who we are at NGI. So at NGI, we provide access to technology for massively parallel sequencing and genotyping at all scales and associated bioinformatics support to all researchers in Sweden. So we offer several types of library prep and sequencing methods, where one of them is de novo reference genome sequencing, which we will be focusing on today. So just to give you a hint on our capacity, so last year we processed over 1,000 projects containing more than 60,000 samples. So this in total generated more than 765 terabase pairs, which is approximately equivalent to producing 1x coverage of a human genome every second minute. So please have a look at our website to find out more on different library prep and sequencing options that we provide, as well as some price information. So you find us at ngisweden.scilifelab.se. You can also find us on social media, so Twitter, YouTube, and Instagram. So with that, I would like to introduce our first speaker, who is Olga Wiener Pettersson. She is a project coordinator at NGI with many years experience on setting up and planning all types of research projects, and in particular the novo reference genome projects. And she will talk about the service that we at NGI can offer you.
So good morning, everybody. Let me try to share my screen with you. And here we go. So thank you for the introduction, Michaela. Indeed, we are at NGI, we're doing lots of different things, but lately de novo sequencing and de novo assemblies of non-model genomes has been one of the main focus areas. So in here I will be presenting in my capacity as a project coordinator at NGI, but at the same time, I'm also one of the two Swedish representatives, the European Reference Genome Atlas Commission Consortium. And shortly about why do we need reference genomes? Approximately 3 or 4 years ago, there's been a huge international effort has been created. It's called Earth Biogenome Project. And within Earth Biogenome Project, scientists have understood that in order to study biodiversity and in order to study biodiversity genomics and evolution, one really needs to have good reference genomes because they offer a completely different understanding of how the genome biology is working and what kind of genomic architecture there is, and also what kind of evolutionary studies can be drawn from the chromosome scale genome assemblies. SciLife Lab is a partner in the Earth Biogenome Project, and we also partner in the ERGA initiative. ERGA is a European branch of the Earth Biogenome Project. And at the moment, it's a consortium of European biodiversity researchers who are interested in genomics. Recently, only less than a year ago, we have, together with a group of Swedish researchers led by Kerstin Lindblad Tull, we applied to VR to try to make the NGI and the SciLife Lab more accessible for the biodiversity genomics people. I will talk you through what are we working with within this project, but it is a 4 years long endeavor to try to make SciLife Lab more and more accessible, not just to medical scientists, but also to the people who are interested in non-model organisms. So our ambition within this project and also our ambition within the next couple of years is to create so-called one-stop shop, where the user or researcher will come to us with a tissue sample, we will perform high molecular weight DNA extraction, RNA extraction, and they will distribute the material into different flows. One would be for reference genome sequencing, that is sequencing the DNA with long-read technologies. Then there will be also scaffolding using the chromosome contact maps. And about that, I will tell you just in a couple of minutes. And finally, to produce RNA sequencing data for the annotation, the functional annotation of the genome. So how does NGI do the genome or de novo sequencing at the moment? Already now we can start with the tissue and we can perform both DNA and RNA extractions. And here a short asterisk and a note that we can do only for de novo projects, unfortunately, due to the capacity, and we're not unable to perform this type of analysis for any other types of the projects where many different samples are involved. So only for the reference genomes, we can extract DNA and RNA. When people ask what kind of quality of the genome do I need? Do I really need to have this super duper reference quality genome? It's everything depends on your scientific question and what would you like to do with it, with the data that you get. We're trying to adopt the Earth Biogenome reference genome standards and working towards the chromosome scale assemblies. Sometimes they also called the platinum genome assemblies when you try to get as uninterrupted genome as possible, phased if possible, and when all the chromosomes or the majority of chromosomes are assembled telomere through centromere to telomere. Also, recently we started a very close collaboration with NBIS. We did work together before, but at the moment we're trying to work on integrated pipelines between our two infrastructures to make a seamless transition of the data from the sequencing machine through the assembly and annotation process so that the PI will get a final product which is completely assembled and annotated genome. It's still work in progress though. So let's start with a scientific question. What kind of quality of the genome do you really need? So we usually ask our users, so why do you want to do the genome sequencing? And two, of course, what is your budget? If you're working with population studies, if you're looking at single nucleotide polymorphisms and Mendelian traits, usually sequencing with Illumina PCR-free libraries and then adding RNA-seq on top of that for annotation purposes should be enough. However, with genomes of this type, you can answer a pretty limited number of questions. So if you want just to make your population screen to look for the bottlenecks, or if you want to look for the variants within your population, that is perfectly fine. However, if you choose to do the chromosome scale assembly, if you want to study complex traits, if you want to make a large evolutionary study on the whole genome scale, then you really need to have this beautiful chromosome scale and phased assemblies. By having a genome of this quality, you're not limited to answering any of the scientific questions that could potentially arise during the scope of your project. So it's basically you're securing the future for your research group. Also, one of the points within the Earth Biogenome project is that we know that sequencing chromosome scale genomes is quite a bit more expensive than the simple Illumina sequencing. Therefore, the ambition of the Earth Biogenome project is to create a global open access database of the genomic data so that people, even from developing countries or countries where there is no access to this super duper compute and sequencing infrastructures, would still be able to conduct research projects of any kind of genome that they would be interested in. So for these chromosome scale assemblies, our Earth Biogenomes projects pipeline would look something like that. We would sequence the DNA with long-read technologies. So that would be either Oxford Nanopore or Pacific Biosciences hi-fi reads. Then we'll do a chromosome scaffolding, and Remy will talk much more about that. And then on top of that, we can do also two different types of RNA sequencing for the functional annotation of the genome. So everything starts, of course, with a sample. So we got a tissue from you, we extracted DNA, and now we have two different technologies to choose. We have either Pacific Biosciences instruments, or we have the Oxford Nanopore. So what is the difference? And what kind of data can those different machines produce? And also, what are the sample quality requirements if you want to sequence on one or on another? And here, of course, the sample will become the first limit. In order to produce PacBio hi-fi libraries, and what is it, I will tell you in the next slide, we need 15 micrograms of chemically pure high molecular weight DNA. There are also experimental low input protocols when you can go in with starting from as low as perhaps around 10 nanograms. And there is also a protocol which starting with 300 nanograms. But these protocols, have to point out, they're very experimental, and the quality of assemblies that you will get, they will be inferior as compared to the classical hi-fi assemblies. So the PacBio instruments, they're present at the moment only in the Uppsala node of the NGI. Oxford Nanopore, on the other hand, is present both at NGI Uppsala and NGI Stockholm. So you could actually choose if you want to do sequencing in Uppsala or in Stockholm. The requirement for the sequencing with Oxford Nanopore in terms of DNA, it's a little bit less. You need around 3 micrograms of chemically pure DNA. But here comes the difference. When you make one library on PacBio and you use 15 micrograms of DNA, that library is enough to be sequenced on 3, 4, and sometimes perhaps even 5 smart cells. In case of Oxford Nanopore, every time you need to introduce more coverage and sequence it deeper on additional flow cells, you need to sacrifice additional 3 micrograms of DNA for every flow cell. That is if you want to make 20 kb insert libraries. But the beauty of Nanopore comes with extremely long molecules. So theoretically, you can sequence completely unshared DNA and get your reads up to a megabase scale. But with Nanopore, the longer fragments you would like to get, the lower throughput you are getting, and also more material will be required. If you want to produce a library with 75 kb insert, we need to go in with at least 5 micrograms of DNA. If you want to go for the megabase scale sequencing, and you are targeting reads that are long above 1 megabase of sequence in length, there you need at least 40 micrograms of pure DNA. And here you can see that I have highlighted two-word combination, chemically pure. Long-read sequencing is a single molecule sequencing. There is no PCR in the step, and you have to sequence whatever molecules which are present in the sample. Sometimes when you extract your DNA from non-model organisms, you can have carryover contaminants from the cell walls or from the extractions. And potentially all those contaminants could impair your sequencing reaction. And instead of getting 30 gigabases of beautiful hi-fi reads, as you would expect from a human sample, you can get as little as 5 gigabases if you're working with some very small and weird insect or very complex plant. Unfortunately, at the moment, there is no good quality check for any of the long-read technologies, which will tell us a priori how well the sample will be able to sequence on the long-read machines. We are working on it extensively together with both technology providers, both Nanopore and Pacific Biosciences. But the bottom line is the DNA must be as chemically pure as possible. That is actually why, because the extraction of chemically pure high molecular weight DNA is a little bit of an art form, and the old art form from 60s, 70s, and 80s, it has been quite damaged during the PCR age when we didn't need to have these beautiful molecules. So that is why at NGI we have started a high molecular weight DNA extraction lab, and we can offer it as a service. However, sometimes, despite our absolutely best efforts, we will not be able to detect the contaminants that are still present in the sample. And even if we do our completely best, we cannot always say that we never fail. So sometimes, even if we extracted DNA, the yield of the sequencing is not stellar. We are getting data, but perhaps not as many data as you could expect if you would be sequencing a mammal genome. So diving a little bit deeper into the technologies. So long-read sequencing, how does it work? In case of Pacific Biosciences, we have very long molecules, we circularize them, and then we sequence the same molecule many, many times. So when you sequence the same molecule, you can align different subreads, portions of the molecules against itself. And at the end, you are getting extremely high quality data. So to make it shorter, with PacBio hi-fi reads, we're getting reads of Illumina quality up to 25 kilobases in size. For de novo genome sequencing, our recommendation is to sequence at least 15x per allele. Oxford Nanopore, it can produce extremely long reads up to megabase size, but the quality of those reads are still inferior to the Illumina and to the PacBio hi-fi. So in case if you are going to work with Oxford Nanopore, we always suggest to add extra Illumina data to polish for the indels. And also in case if you're working with Oxford Nanopore, at least 20x per allele is required, but 30x per allele is recommended. To continue with that, after we've done the primary assembly, we can also improve it by looking at the arrangement of the DNA molecules in the chromosome, and that is so-called high-C or omni-C method. So if on the bottom of the graph, you see the sort of big globules or big knots of DNA. So those are the nucleosomes. So using this protocol, we can understand which DNA molecules were found on the same chromosome in the same nucleosomal complex. And this information can be later mapped to the PacBio or Oxford Nanopore assembly. And we'll sort will serve as extra sorting of the contigs and also assigning the contigs to chromosomes. And my colleague Remy will go much deeper into the details and tell you how the workflow works. And at the moment, this technology is available only at NGI at Stockholm. As Ignas is absent today, I took the liberty to add a couple of the slides that he would have covered in his presentation. So I will show you a little bit of the tree of life projects that we've been working with. So just in the past year, we sequenced at least 200 non-mono eukaryotic genomes, and at least 60 of them, they were done at the chromosome scale. The largest genomes that we sequenced so far, there were 2 different conifer species with 22 gigs. We have also sequenced a salamander, and we also sequenced Arctic krill with a genome size of 18 kb. And on the small scale, we've also been sequencing some yeast samples, and we've been sequencing insects with genome size of around 150 and so on. And here I would like to give you a sort of a sneak peek of what those hi-fi reads can do for you. And here is an example of a bird project that we have, and we sequenced it on 2 SMART cells. So we generated quite a lot of the data, as you can see. And also the quality of reads were really good. If you look at the graph on the right side of the screen, you can see the number of bases that are of Illumina quality and higher. And when you do the assembly, we can offer that as a service. We can produce the draft assemblies here at NGI. When we're talking about the PacBio assemblies, before we give it to Remy for the high-C scaffolding, we are using different algorithms. We can use the PacBio's native IPA, or we can use Hi-Fi ISM. They produce very similar quality assemblies. They have just different requirements for the compute infrastructure. But the bottom line is that you are getting completely phased assemblies. So these assemblers are able to disentangle large chunks of the chromosomes. But sometimes we have very interesting surprises. If everything works fine with mammals, vertebrates, and birds, sometimes with insects and plants, that's not exactly the same. So here is an example of surprises that you can get when you do de novo assemblies. So for this genome, we thought that the genome size was 300 megabases. Then we sequenced that, and we tried to assemble it and look at the quality, and we found out that the genome was extremely fragmented. So we tried to make a prediction of the genome size estimate, and it turned out that the genome was at least 3 times as big as it was supposed to be. So instead of 300 megabases, the genome actually turned out to be 1.7. But at the end, we were able to get very nice assemblies. When it comes to the RNA sequencing, we can also do that for the annotation purposes. The most common way we do the poly-A selected mRNA, and there we recommend to sequence that from several different tissues. Also, we can help the annotation process by adding RNA-seq with long reads, where you can produce the entire transcripts in one read. That can assist the annotation, and Henrik will talk more about that. Also, you might consider sequencing other types of RNA, like microRNAs, long non-coding RNAs, and so on. And these services, they are available both in NGI Stockholm and NGI Uppsala, but microRNA is mainly done in Stockholm. So we are working on the entire package with assembly and annotation. What you get from NGI after Ignas done his job and Remy has done his scaffolding, we produce a very raw draft assembly, and quite a bit of work would be still required. So for that, we would either leave the data to the research group, or we could give it to NBIS to do the manual curation and polishing of the assemblies. And after that, the NBIS professionals can go in and do the annotation. So from a single organism, you will also get a functional understanding on what is going on there. So how should you proceed? You should first start with visiting our homepage, because there you can read much more about what our services are and how they are functioning. You can, if you're unsure about the project, the setup, and so on, you're most welcome to request a meeting with us. And then finally, you can place a portal order. So speaking about the homepage, I can just highlight 2 things. All the services that we have, they have a small subpage when you can read the description of the methods. And also there is information with some price examples and also how to prepare the samples and how to send them to us. And if you want to place a project in the portal, here is the snapshot and there is a link. It's accessible from our NGI homepage. And you can either request a meeting that is on the top, or you can also request a de novo reference genome sequencing project. This form we have launched this Monday, so now you don't have to apply for 25 different projects that you want to do, like RNA sequencing, DNA extraction, and other types of sequencing. So all of that can be now submitted within a single submission form, and then we'll get back to you with more information. So with that, I would like to stop my presentation. And I would like to stop sharing the screen, and I will see if I have any questions.
Thank you, Olga, for that nice presentation and for cover up a bit for Ignaz. I think that if we have a few questions, but we can actually answer them in the Q&A chat, or we will bring them up later on at the Q&A session. So I think we can introduce our next speaker, who is Remy Olsen, and he is a bioinformatician at NGI, and he will give us an overview on how high-C methods can be used to generate chromosome scale assembly scaffolds. So the stage is yours, Remy.
Thank you, Michaela. I will share my screen. There. I guess this looks fine. Okay. Yes, so I will talk about high-C and how it can be used in scaffolding or assembly scaffolding. So first let's just talk briefly about the method or chromatin conformation. This is a very high-level generalized overview of the method. So in the nucleus, the chromatin is sort of packed into different chromosome domains, and it is different sort of 3D structures. So the aim is to capture these structures, and by doing that, you cross-link the chromatin, and you sort of shear the DNA on both sides. So just capture the crosslink. The blue and red sides are the different sides of this crosslink. Then you ligate the ends, and then do reverse cross-linking. So you get these fragments that can be worked on to create a sequencing library. And of course, we will sequence them, this library. And I'll just also say that this digestion, or sharing, it can be using a single restriction enzyme, multiple restriction enzymes, or DNase. Right. So since I'm a bioinformatician, the way I think about it is to map reads and look at the distance between read 1 and read 2, what you might call insert distance also. So this is just a typical library that was mapped to a reference genome. And you see most of, or a majority of the reads, about 50 million, are up to 1,000 base pairs long. But you see a very long tail. I mean, this is above, or this here is between 10 millions to 100 million base pairs. So you get some very long reads, or long, or very much structural information about the genome. So what this can be used for, I will just, before I get into scaffolding, just mention 3 use cases. I mean, one is epigenetics. What you see here is the way that DNA loops around itself when promoters and enhancers interact. So this can actually almost be read from the, or this can be visualized. I will get to that later, given that you have a high enough resolution of data. The other thing, as I said, this gives also structural information about the genome. So you can use it for like SNP phasing, and you can also do structural variant calling with this data. So at NGI, we have two sort of kits from two commercial vendors. One is Dovetail. It's the OmniC kit, and it uses DNAs. And the other one is from Arima, which uses multiple restriction enzymes. Right. So once you have sequenced this library, what you will do then is, or what you should do then, is to do quality controls. So there's several tools available for doing this. I'll just mention a few: Juicer, Pair Tools, and HiC Pro. But these tools have much in common with each other. So the common tasks include read mapping, deduplication, filtering of ambiguously mapped reads, and then also rescuing such reads and rescuing chimeric reads. So I'll just give you a little bit of an example of what these programs might do. So this is just taken from Pair Tools. So here you see a one-sided mapping. Read 1 doesn't map to the genome at all, only read 2. So I mean, this doesn't really give us any information, so it might just discard it. And it can be multi-mapped. Read 1 maps to like several positions in the genome. So that's a little bit problematic, but perhaps if you say that one of these mappings are very low quality or very, it maps not with a high confidence to one place, then maybe you can do something about it, and some tools do that. And the third one is chimeric reads, I would call them. So you see that read 1 is sort of comprised of two sort of genomic locations. So what you can do here is that if let's say that this end here, the green end here, is very closely to the blue end, so that they might be from the same fragment, you can perhaps discard it. But there is several strategies for doing this and making these decisions, and these tools have several. I mean, they have different strategies. So just to keep that in mind. So also, this is for Pair Tools, so you will often get like statistics out from this process. One more important one is like percentage of the reads that are non-duplicate. They are cis mapping, so they map to the same chromosome, and they have a distance between each other more than 1 kb. So this is the most important stat that you can have. So these are basically the reads that go into the scaffolder, right, after pre-processing. And we would like that to be like, you know, this portion to be somewhere between 60 or 80%. I mean, that would be normal. And of course, you can look at the percent of duplicate reads. I mean, sometimes we do like a test sequencing just beforehand. So, I mean, you have to and estimate library complexity, so you can sort of know what you will get out of the library beforehand. And also the percentage of map reads. So as you saw in the previous slide, some of these tools will just discard reads, you know, when it doesn't give any useful information. So this can be, you know, unmapped reads that it can classify it as. But yeah. And also for quality or QC, you can visualize the mapping. So I will just show the output from a software called Juicebox. And I will use this output from this program many times in this talk. So it's a method for visualizing high-C data, and it also includes functionality to sort of reassemble genomes. So what you are looking here is kind of what the plot that Olga showed you, but just much more zoomed in. So what this is useful in QC is that you can sort of look for these sort of telltale patterns of compartments and chromatin interactions. It's just to check if that you have saturated the library, right? Or yeah, and that it can be useful. But yeah, I will come back to this. And then I also just mentioned the scaffolders programs that are out there, a few of them. So you have 3D DNA. That one works with both Juicebox, as I showed in the previous slide, and Juicer, because it's made by the same people, right? So the inputs and outputs of this program go into Juicebox and Juicer. Another one is also 2, and the third one is HiRISE. So this is a dovetail tool, but it's not available for anyone to use. So Dovetail has a service. You have to submit your data to them, and they will run it. So hopefully one day it will be available to anyone to run it, but not currently. So I would also just say that this program is developed by or rather that the developer of SASA2 has been hired by Dovetail to develop this HiRISE scaffolder. And the last one is Instagram. So this one is based on GPU processing. I have not used it, and we will not at least provide this currently. So the scaffolders we use are 3D DNA and SASA2, right? So yes, now we'll go through a couple of examples of assembly projects. So the first one is the Paradise Crow. So this one is with Alexander Suh, who will be speaking later. So first about the input assembly for this project. I mean, it was sequenced on PacBio and assembled with Falcon. And also 10x linked reads was used for initial scaffolding. Then also Dovetail Chicago. So Dovetail Chicago is a method that is kind of similar to Hi-C, but on a smaller scale. I will not go into that one, but it can be used for scaffolding as well. And this is also another method that only Dovetail provides as a service, not as a kit. And at last, also phase genomics Hi-C. So this was used for a very conservative scaffolding, and also several rounds of manual creation and polishing till you get a very nice and polished assembly. So N50 6.6 megabases. So N50 is it tells you about how the contiguity of the assembly. Same with L50, so it's 50 scaffolds. So if you can say that half of the genome is contained in only 50 scaffolds, right? So this was one of the first, or it was actually the first Hi-C project that we had at NGI. So I mentioned the phase genomics. This was submitted to phase genomics, the material, and sequenced at them. But what we evaluated was at NGI was Dovetail Genomics kit and the ARIMA kit of the Paradise bird. We also had at the same time material for another bird. I think that was also an Alexander Suh project. We ran 3D DNA for all the Paradise Crow samples. And what you can see is that from the sort of traditional assembly metrics, they look very much the same, right? N50 is the same for all of them. L50 is 6. That's kind of interesting. But yeah. And then if you look at the juice box map. So I will just briefly explain that these blue boxes, they are the borders of the high-C scaffolds, right? So then it's very like tempting to maybe say, oh, maybe these are chromosomes. But this is just what we see at NGI. Of course, I don't think I will spoil any of Alex's talk by saying that they were able to find our chromosome model from this data, though. So, yep. But I will come back to some more details about this. So the second project, and I will be very vague here because this is a very ongoing collaboration we have. It's a plant genome. It is very large. So it was first sequenced to 50x on PacBio. It was assembled. It was extensively haplotype per. So I will get back to that actually. And it was very sort of polished, and other curation steps was done here. We sequenced or prepped and sequenced 4 dovetail high-C libraries, and these were pre-processed using Juicer and then scaffolded using Salsa2. I'll also say that they had a genomic map of markers with about 20,000 markers in it. This will also, yeah. So this is showing 4 chromosomes that was, or what we think are 4 chromosomes. This is just to show that, yeah, it is possible, right? I will actually use this one for other examples in later slides. Okay. So I will not only just brag about this method, so I will also point out some problems, right? First one is handling tissues. So this is just taken from our website, ngisweden.se, for the dovetail OmniSeq kit, right? So it has very sort of these are just the amount of sample, the input amounts needed for this prep, and this is I mean, we don't extract any DNA in this prep, but there's like requirements for sample storage, like it needs to be snap frozen in liquid nitrogen, for example, for tissues, and there are requirements for we will ask you to grind the tissues. Also, there are correct ways and wrong ways of doing this as well. And also, perhaps most importantly, do not thaw or avoid thawing these tissues, because that can be very catastrophic. So I just want to stress this point. Yeah, very hard. Okay, so if you look at some of the high-C maps, right, or the contact maps, this is again in Juicebox, we can look at some subtle issues with high-C scaffolding. This is again looking at the paradise crow, sorry. And again, here you see the blue box. This is a high-C scaffold. The green boxes, they represent the borders of the input contigs, right? So the PacBio contigs that was used as input for the scaffolding process. So what is wrong here? So yeah, it's an inversion. It's very subtle to see, but there is a sort of a diamond pattern here. So you can see this edge here, and you see also here there's a sort of a discontinuity here. So the thing about high-C, of course, is that most reads are, as mentioned earlier, are the majority of them are shorter than 1 kb, right? So you would expect most reads to be centered around this diagonal. So you can almost think of this plot as a dot plot, right? Where further you go from the diagonal, there you will find high C contacts that have a longer mapping distance. Okay. So yes, in Juicebox, this can be fixed by just a single click of the mouse button. It can look something like this instead, right? Second one is scaffold borders. So here you see in the middle, you see a very like densely packed area with high C contacts, but there are two scaffolds here. There should be one, and this can also be easily fixed. Then you can have something like this. I mean, it's not very, a lot of the issues with high C is that it can be very ambiguous. Like, what are you going to do here? Are these 1, 2, 3, 4, 5 chromosomes? Or is this one big chromosome? And these are just local compartments. Yes, so that's, you can encounter things like this. So I will also just mention another sort of feature from, this is from the plant. And this is something that can be, that you can see. So what is this? So you see that there's two like sequences here that are next to each other, but there is very few contacts that link them together, right? So what we think this is, these are haplotigs that are, you know, assembled in tandem, right? Normally these two would be merged if you have a haploid representation of a genome, but for some reasons, I mean, these two was not merged into one sequence, but rather stuck one after the other in the sequence. This is, yes, so to aid you in this sort of process, it is very helpful to have other types of long-range information from, you know, optical mapping, physical mapping, or just synteny information. So this is showing one of these chromosomes from this plant project, and these are a map of markers, right? So before and after the high C manual creation steps. So the red dots are the markers before, and the black ones are after. So, I mean, for instance, here you see that this is most likely an inversion that was fixed, right? You don't see the black ones being across the diagonal. Right, okay. So to summarize, I mean, chromosome scale scaffolding is possible using high C. But in my opinion, the bioinformatic tools that exist today, they are very immature. So more time will need to be spent developing this. And time and attention is required for polishing the initial draft scaffold that is produced by us. And I think that's everything I wanted to talk about.
Thank you very much, Remy. So I think you have some questions in the Q&A chat that you can either answer directly or you can save them for the Q&A session.
Yes.
So the next speaker is Henrik Lantz, and he is a joint head of facility support and infrastructure at NBIS. And he will give us an overview on who NBIS is and what they do. And he will also tell us how NBIS can support you in assembly and annotation projects.
Thank you, Michaela. I will try to share my screen. All right. Great to be here. So my name is Henrik Lantz, and I work as a support manager and as a joint head of facility for the facility support, infrastructure, and training in the bioinformatics platform NBIS. So I will talk to you today about what we can help you with when it comes to genome assembly and genome annotation. So if we look at the Scilab lab structure, we have the bioinformatics platform up here, with which is exactly the same thing as NBIS. And then we have genomics here, where NGI, where Olga and Remy already have presented. And I am working then specifically for this facility here, support infrastructure and training. We're a fairly large platform with around 100 staff. We have around 60 bioinformaticians working, and these are placed all over the country. You can find us in 6 cities in Sweden. And with these 60 bioinformaticians available, I think I can promise that we can support you in basically all your, at least, sequence related bioinformatics need. So we work mostly with DNA sequences and RNA sequences, but we do also have some competence in proteomics, metabolomics, statistics, and so on. But the bulk of the projects we see definitely deal with sequence data in one way or the other. We also have a large system development team that can help you to do, more programming, develop tools, and so on. We have a big data management team that can help you with data management plans. And we also have people working for us in compute and storage that take care of the clusters that I think many of you are using at the Uppsala, for example, the Rackham and Bianca clusters. So more specifically, the assembly and annotation service, we're currently 4 of us in this team, but we do have a 5th member joining us in October. And we've been doing this since 2013, where we mostly are working on annotation, but we also have a lot of assembly competence. So we have our own cluster where we do the analysis, and we work on all types of organisms. I would say we've covered all the major groups in our 8 years we've been doing this. Just added 2 publications that we've been doing the annotation for. So the paper on the wall listed there and the basket willow, just 2 examples of projects we've been involved in. So if we go then to genome assembly, and here I will focus on validation and curation. This is how I imagine a typical workflow for a genome assembly and annotation project would look like. You start with planning your project, and then you extract DNA and RNA for use in annotation, and you choose the technology you want, and then you go into sequencing. Once you have the data, you need to see if the data is any good, and then you go into assembly. But once you have the assembly, then is that assembly any good? You might want to try different tools. You might want to compare different assemblies. And then you might want to correct any problems that you find. And once you have an assembly that you think is good, you want to do repeat masking and then go into annotation. And in the first part there, we find it really, really good if both NGI and the bioinformatics platform are involved. So at the first meeting where you want to discuss and plan your project, we like to be present, both of us. But in practice, I would say it's now NGI that does this first part, of course, the lab part. NBIS does not have any data producing capabilities. We're only staff. And then, for example, then Ignas and Remy that I presented today will take you all the way up onto the assembly stage. And then we tend to take over in the more downstream types of analysis. Although we could also be present and help out in all of these other stages also. So if we talk about assembly validation, the questions you want to ask yourself there, okay, so I have an assembly, but is it any good? Are there any contaminants in there? Are you perhaps working with an organism where you might expect there to be symbionts? I mean, if you're working with a marine organism, for example, you can find many, many more organisms in the tissue perhaps than the one you're actually interested in. Are there any misassemblies? And how much of the genome can actually be found in the assembly? Have I'm, looking at genes, for example, all the genes I can expect in the organism, do I also have these in my assembly? So some examples of things we can do is, for example, we can do something called k-mer analysis, where we can see how many of the reads that you have input into the genome assembly tool are actually used in the genome assembly. We want a situation on the right there, where most of the space under the curve is actually used. We could also look at the assembly graph. So the assembly tools will take the reads and create a graph and then create this assembly for you in the end. And one way of troubleshooting is actually looking at this graph, visualizing it and see how then the tool has actually used this to create the assembly. And on the left there, you have what would be an ideal bacterial assembly, for example, one circular chromosome. On the right there, you will have something that is much more complex. We can look at contamination in the assembly using Kraken, for example. This is example here is from bacteria, where you can find a lot more bacteria than the one you actually thought was in your sample. Another way of visualizing that is using blob tools, where you would like to have one blob basically, and not two like you have in this case, where you have one large elongated blob up here and one smaller here. So these are the contigs that are then plotted based on GC content and coverage, and then these contigs are then blasted. You do a blast search and see what kind of organisms hit these different contigs, and the different colors you see there would actually be different organisms. So here you have more of a mix than you would actually want to, and you have two separate blobs. So this is an indication that something is a bit off. If you have a genome that is closely related you want to compare to, you can align the genome to that reference to see insertions, deletions, and all sorts of problems. Some of these could be biologically real, of course. So there you have to verify that in some way, but they could also be misassemblies. If you're working with bacteria, you can do this multiple alignments. It can also be done with some other organisms, but mostly used for bacteria, where you can see, compare the genomes and see how the different sections have perhaps moved around, or if there are any misassemblies, you would also spot that here. And then assembly curation. Once you have identified problems, you also need to correct them in some way, and this we can also help you with. And in some cases, you might want to do polishing, so basically removing problems in the assembly by using another sequence technology, perhaps Illumina reads. This is much more rare today that this polishing is needed now that a lot of people are using PacBio hi-fi reads, but for Nanopore, this would still be, I think, something you want to look into. So, whoops, the subtitle there is wrong, but if you look at genome annotation then. So once you have your assembled genome, you basically have something that's called a FASTA file that you see on the left there. So a long stretch of nucleotides without any extra information. So to figure out where in this assembly you have the genes, for example, and what they do, you need to do annotation. And in your downstream analysis later, if you want to do like differential expression or something like that, you need a genome and you need an annotation file, usually in the GFF format. And this is what the annotation process will result in. So to the right there, you have a screenshot from a genome browser, which is often the result that you're interested in, where you have several gene models. You see all of them go from right to left. And on the bottom of the slide here, you also have an example of what a GFF file will actually look like. So the main steps in genome annotation first involve the QC stage, quality control, and then we move into structural annotation. So this is figure out where the genes are in your genome and what they look like, how many exons and introns are there, and so on. And we use a tool called Maker to do this. Then there might be a step called manual curation, because the annotation is a computational process. And even though these tools are good and we do our absolutely best with it, there will be problems still in the annotation. It will not be perfect. And but you might yourself have a lot of knowledge about some genes that you're interested in, for example, And what you can then do is manual creation using a tool called Web Apollo that we also supply to you, where you go in and change the genes that you're most interested in yourself. And once we have the structural information of all genes, we can go into functional annotation where we figure out what these actually do. So what function the gene has. And this is done by mostly a tool called InterProScan, but it can also be done by similarity to known proteins from other organisms. And then you want to move into downstream analysis, and you also want to submit your genome. And this is also something we can help you with. So just a little bit more detail. The tool we call Maker is a pipeline that combines different types of evidence to do the best possible annotation. So it uses ab initio models. This is what this column represents. So basically tools that find genes in your genome based on likelihoods. We can use proteins from related organisms, protein sequences, and we can use transcripts. And this is really, really awesome. Sometimes it might even be possible to have RNA-seq reads from the actual individual that the genome used in the reference genome was extracted from, and that's brilliant. But often it might be okay with closely related individuals. And this will give you a very, very good idea of what the genes look in your organism, and also RNA-seq will give you a good chance of even going into isoform if that is of interest to you. So this is just an example from Web Apollo, the tool we use for manual creation that we'll make available to you, where you have some protein sequences aligned here, and up here you have the final annotation. And these boxes here, you can then just extend, shorten, you can add exons and so on quite manually just by dragging. So this is a graphical interface that's really, really useful. So to summarize, so we use Maker, which combines ab initio gene predictions, protein sequences, and transcript data to give you the best possible annotation. When we do this, we want a frozen assembly. So if you turn to NBIS and have already done all the work leading up to this, to the annotation, remember that if you go back and change the assembly, we might actually need to redo the annotation from scratch. So we prefer that you're absolutely finished with the assembly when you come to us, and we call this a frozen assembly. We can supply Web Apollo for manual creation. You will receive a GFF file fully standardized to use in all of your downstream analysis, whether it's gene expression or population genomics that you're interested in. We can also help you to submit the assembly and the annotation to a European Nucleotide Archive, which is what we prefer, but this will then also be mirrored in GenBank. And we are also available for all sorts of downstream analysis. So just briefly, a few minutes then on the support models we have and how we can help you. You can always come to us for a free consultation. We do this a lot, and it's completely free, and it's usually handled in the form of a meeting. So you come to us, you file a support consultation request, and then we put the best expert for your project. We sign the best expert for this project, and you have a meeting. We also have something called the short-term support model, which is where there's an hourly fee. We do a lot of annotation projects this way. We also have something we call long-term support, which is free, but this one has application rounds. So 3 times a year you can apply to get this free support, and this is possible by funding from the Knut and Alice Wallenberg Foundation. There will be a scientific committee that evaluates all applications, and then the best, around 20% I think in each round, are picked for this free support, which is 500 hours for, and will probably run over 2 years. So this is really, really good support if you can get it. So, and I really encourage everyone to apply. There is also a mandatory involvement from the research group in this support model. So there needs to be someone in your team to act as a recipient for bioinformatics knowledge. So this is important to remember. Could be a PhD student, postdoc, etc. We also have something we call partner project, which is calendar based. And so if there's a really big bioinformatics component in your research, you can then perhaps this partners project model would be good for you, where you can have one of our experts for perhaps 50% of that person's time over 2 years. You get support by going to our webpage, nbis.se, and you press the support on top there and apply here button, and this will get you to a form where you can, register a consultation or support or a partner project or long-term support request. Just very briefly, I also wanted to mention that we have something we call drop-ins. So every week online now, Tuesdays at 2, I'm sorry, that is actually incorrect. We do have drop-ins. So all sorts of bioinformatics questions are welcome. We have perhaps 30 or 40 bioinformaticians waiting for you to answer your questions every week. So very welcome to join this. And you can find the Zoom link at the events page on our webpage. So thank you so much for listening. I would be happy to take any questions.
Thank you very much, Henrik. So you have one question in the Q&A chat that I think that you can answer directly in the chat.
Will do.
Thanks. All right, so last speaker of today's session, but definitely not the least, we have Dr. Alexander Suh from the University of East Anglia, UK, and Uppsala University. So Alex's research focus is on studying evolution in animals, transposons, and viruses using comparative genomics, molecular biology, and phylogenomics. And today he will talk about how he has used the NGI research infrastructure to study sex-specific and tissue-specific genome differences in birds. So thank you, Alex.
Thank you so much. Can you all hear me? So I'm going to share my screen now, and I just want to say thanks so much for having me here and giving me a chance to speak about some of the work we've been doing. And maybe also highlighting that, well, there's so many things we can do now with all these amazing technologies that were featured earlier today. And I kind of try to wrap up this session by focusing on some of the things we're interested in our group, kind of focusing really on the gaps, the tricky bits of the genome, and then what biological questions we can ask and hopefully also answer based on that. And really the focus being, first of all, on sex-specific genome differences and tissue-specific differences. And just to get us all on the same page, what does that mean? What do I mean with that, basically? The first thing is really, let me fix this pointer here. The first thing is that when we talk about sex-specific differences, I really mean the sex chromosomes here. So in mammals, it would be the XY system, so Y would be the male-specific chromosome. Here in birds, what I'm going to talk about today is going to be the female-specific chromosome, so that is the W chromosome down here. And then in the second half of my talk, I will talk about tissue-specific genome differences, meaning that in some cells of the organisms, you have a specific set of chromosomes. In other cells, you have those same chromosomes plus an extra part of the genome, basically, that only exists in this case in the germline. And I'll explain what that means and how we can study those different parts of the genome. So let's delve into this. The first thing is really part of our efforts to understand the evolution of birds of paradise. I think I don't have to motivate why it's interesting to study these cute little birds, but the idea really being we are interested in especially transposable elements or repetitive elements in general, in particular on the W chromosomes, of the female-specific chromosomes. And I just want to get us all on the same page with one slide to kind of explain what we expect to find here, just to kind of also know how we have to set up our sequencing experiments. The idea being that at any given point in time, we assume that sex chromosomes stem from an ancient kind of autosome pair that is fully recombining. And then you have a sex-determining locus here that if there is suppression of recombination together with sexual antagonistic locus, this then is by definition a sex chromosome. And so if over time this suppression of recombination proceeds further and further stepwise to get these different strata across the sex chromosomes, we end up with a situation that, for example, here most birds have nowadays. So you have basically a male recombining Z chromosome that more or less looks like a normal autosome to some degree, at least relatively spoken, hundreds of genes in there. And then the W chromosome, which is mostly non-recombining and highly enriched in repetitive elements. and looking quite different from the Z or the ancestral autosomal pair by only containing maybe 2 or 3 dozens of genes, basically, at least in birds. So the idea really being, when we have this in mind, we need to really expect the worst in terms of our assembly efforts and need to really assemble those bits of the genome quite well and hopefully trust it at the end, right? And Remy was talking about this earlier, so I don't want to repeat too much of that. He was basically using the Paradise Crow here as an example. So this is part of this project I'm talking about today. This is led by Valentina Peona, my PhD student. And she's basically spent a lot of time evaluating all these different sequencing technologies that we generated together with NGI, but also together with Dovetail and Face Genomics back then, because this was kind of the pre-Hi-C service area, basically. But the idea really being, with all these technologies together, each of them have their benefits and each of them have their problems, and we need to go through these manually and curate them to have an understanding of what is used for what different parts of the genome, basically. And I can highly recommend having a look at this paper that just came out now in Molecular Ecology Resources, where Valentina really lined up the different lines of evidence for each single gap in the genome to understand what do you want to study and what do you need to do so in order to have a nice assembled chromosome level assembly. And I guess lo and behold, I just want to say, I think we are at this point now also then at a kind of erga quality chromosome level assembly. So despite the issues that also Remy was mentioning with the Hi-C, there are actually quite some reliable aspects of how the chromosomes are scaffolded, at least in terms of the overall synteny. And I just want to kind of hint at a couple results here. But maybe kind of a take-home message of what I think where we are at now in this current third generation era of genomes. If we think of a true genome or a true chromosome, like this would be everything that we have in nature. This would be the telomeres, then we have the centromere here, lots of repetitive regions, lots of genic regions, of course not to scale, just a kind of cartoon. We are able to study most of these single copy regions and many short repetitive elements quite nicely with short reads. I think that's well possible. But some of the other more messy bits we can study now with long reads, and then with different scaffolding technologies, we can either add these to assemblies or at least put them in context. So we have a rough idea. Let's say here we know now where the centromere is. We still don't know how the sequence maybe looks like in total, but we get to the point where we have a rough idea of many of these previously inaccessible genomic regions, especially repetitive regions, that we can now study. And I really just want to focus now the remaining slides of this part of my talk on just the sex chromosomes, just the W chromosome, because I think maybe this helps to kind of appreciate the beauty of these different assemblies. So again, back to the Paradise Crow, we expect them to have a very small or very repetitive W chromosome and the kind of normal looking Z. And I give it away in the title, the W chromosomes are very highly retrovirus enriched. The importance being we have really also done a lot of annotation, manual annotation of the different retroviruses and transposable elements here. But the idea being, when we now extend this to other species, so this was together with postdoc in the group, Julie, and data we were able to use from the Vertebrate Genomes project, so also part of the ERGA initiative. These different birds, 6 different birds with similar assembly quality now, and different chromosome states. These have very divergent or differentiated sex chromosomes, and emu has not so differentiated sex chromosomes. We actually end up finding more or less the same pattern, that all of these W chromosomes are highly retrovirus enriched. Just want to kind of show this in these little pie charts. Just have a look at this one here. In black, we have the length, the total length of the autosomes. Then in gray, we have the Z chromosome. And then this tiny part here is our W chromosome. So this is something that makes up less than 1% of the assembly. It's a tiny part of the genome. This looks more or less the same in the other species as well. But when we look at full-length elements, so full-length transposons and retroviruses, these are basically those parasites that are able to make more copies of themselves in the genome and have deleterious effects on nearby genes, for example. You see that these are basically more than half, if not 2/3 of the chromosome, and even in emu, 1/3 of the total elements are just on the W chromosome. So the idea being, we not only have a chromosome that's packed full with different repetitive elements, but those that can make more copies of themselves elsewhere in the genome are all enriched on this one chromosome. So it's a female biased or female specific enrichment of these elements. So we kind of thought maybe this is something we can test further with some predictions in the future. And we came up with the idea that maybe we can kind of measure this as a toxicity index. This is related to a concept of the toxic Y by Doris Bachdrup, where there's experimental evidence that having a Y chromosome is really bad for the fly. So we can't do this for birds, but the idea being, what if at least we can measure the excess of intact repetitive elements, those that are able to make more mutations in the heterogametic sex, here the females, over the homogametic cells, here the males. And this is basically something that really varies. We have rather expected lower toxicity basically in the emu, for example, but rather high, for example, in the hummingbird. And it's something that hopefully we can now test in the near future with experiments and at least looking at expression and or transposition of these different elements in males versus females. Kind of as a thought at this point, I just want to leave you with the idea that what does it mean, toxicity, in this context? So the idea being, if you're a male bird, you just have autosomes and Z chromosomes, which are relatively devoid of repetitive elements. That's not that many intact elements. Whereas in females, you have the same chromosomes plus an extra one here, half of like just one Z, and then this W chromosome, which seems to kind of radiate repetitive elements elsewhere in the genome. So what if that could actually lead to some observed differences, for example, in lifespan in males versus female birds, because female birds actually tend to live less longer. This is of course completely speculative at this point, but a thought to test in the future with the evidence that we have now for these different transposable elements, because we can now measure their expression. And kind of a thought in terms of the Haldane's rule. Haldane's rule meaning that if we have hybrids between two species, the infertile one will usually be the heterogametic sex, so in birds the female. Could that actually be partially at least caused by the divergence between W chromosomes? So you have regulatory differences or misexpression of specific elements that can have negative effects here. So much for that. I just wanted to kind of put these in context here because I need to kind of conclude that part of my talk. I just hope I could convince you that we now have the means to study evolution of W chromosomes. We still have lots of gaps in that, but I think we have an overall of the idea of the euchromatic parts of the W. Seems to be rapidly evolving. And more or less always when we look at birds, at least highly enriched in specific repetitive elements, especially intact endogenous retroviruses. And so now at this point we wonder, okay, does this mean there's a female bias in mutations? Does this mean, for example, there's sex bias aging attributed to this phenomenon? Again, this is still speculation at this point, but we hopefully can test this now because we have the data and we have assemblies to map against. And the way we are following this up now is basically to trace the evolution of the W chromosome across different timescales, and then also to trace the mobilization of retroviruses in different stages of gametogenesis in males versus females to see, do we find those virus particles expressed and do they make more copies of themselves? Okay. With that, I hope it's okay if I just jump to the next part of my talk, because maybe that's a bit more surprising results than I suspect many of you have kind of thought about sex chromosomes in that regard before. The second part of my talk will be about germline soma genome differences. This is not in birds of paradise, but this little guy here, the zebrafinch. And the idea being that when we think about what we learned in high school already, that we are multicellular organisms, but we start with a single cell in our development, we expect kind of more or less our genomes to be the same in all different cells, right? Plus, minus, but that's not always the case. There's a bunch of different organisms, both in different, for example, insects and different nematodes, as well as different vertebrates, including birds, that have actually differences between the genomes of different tissues, and especially between the germline and soma. And I want to just kind of simplify that as a cartoon. This whole phenomenon is called programmed DNA elimination. The idea being you can have something that's called chromatin diminution, where you have different parts of chromosomes that are, in a sense, spliced out, chopped out during development, so that in the germline, the chromosomes look like this, and in the soma, it looks like this. So the chromosome morphology changes a bit, and some genes are thrown out. And this has been quite well studied, especially in ciliates, but also some nematodes, for example, and also in lamprey. So quite a fascinating phenomenon where the genome sizes and genome arrangements can be significantly different between different tissues. And the second aspect here, which has been relatively understudied, are so-called germline restricted chromosomes. So this means there's a set of regular chromosomes, or A chromosomes, as I will refer to them in the rest of this talk. They exist basically in all cells of the chromosome. These are basically the shared chromosomes. And then we have an extra chromosome or extra pair or multiple pairs of extra chromosomes that only exist in the germline and then removed from the soma during development. As I'm going to talk about today, this is known from songbirds. I will talk about what the evidence is for that, but there's also some evidence for hagfishes and also for dipteran insects, some species at least in those groups. Okay, so let's delve into this. This is not something that we found. This has actually been, for birds at least, known for quite a while in a beautiful cytogenetic study in the late 90s, looking at this guy here again, the zebrafinch, and then maybe 10 years later also they found the same pattern in Bengalese finch, a close relative. The idea being, if you look at somatic cells of the bird, so body cells, the mortal cells basically of the bird, you'll find here now the 8 biggest chromosome pairs. There's another 32 more pairs, but the point being, males and females differ only by Z and the W chromosomes basically. So that's I guess as expected. And now they took the same bird, but the different tissue, the testes in this case, as a germline representative, they find the same chromosomes, the regular chromosomes, plus an extra chromosome, which happens to be the biggest chromosome of the karyotype. So that is bigger than this chromosome here, which we know to be 150 megabases in size. So 15%, 15% of the genome being made up of this extra chromosome that was up to that point basically very mysterious and also not included in sequencing efforts. And so up to this date, when we started this project around 4 years ago, what was known instead was mainly based on cytogenetics, because it is such a massive chromosome that people can study that quite nicely under the microscope. And so the idea being very, very simplified summary of what is known from that perspective, it is not inherited through the male line because it's eliminated from during spermatogenesis. It is inherited through the female line, so the oocyte will have that. Then there's a big black box of embryogenesis where we don't actually know what's going on at all. And we just know what the adult state is basically, where we have soma absent for this chromosome, male germline 1 copy, except for the sperm, of course, female germline 2 copies, and then soma again absent in females from that. So this is a rather fascinating state, and something where, of course, we now need to think about how we best sequence that. So, but back then when we started this project, we were scratching our heads, and just happened to talk to NGI back then, who had just established basically 10x linked read sequencing as a technology and as a service basically. And I just want to emphasize this is work that was started by Cormac, my first master's student in the group basically, and also then since then continued by Francisco, who was a collaborator back then and is now a postdoc in the lab. But in a nutshell, what we did here was to sequence 2 genomes for the same bird. So the idea being, if you want to find tissue-specific differences, you need to make 2 genomes. The idea being here, we use liver as a soma representative and testes as a germline representative. And I know that 10x Chromium is unfortunately now not offered or discontinued as a kit, so it's not being available anymore. But I just want to emphasize the way this linked read technology works is that you get basically Illumina short reads, very accurate reads that have a barcode information of where in the input molecule they come from, because you have this microfluidic library prep. And as a result of this, you need only very little DNA amounts per tissue, and you can then make a de novo assembly and have these long haplotypes also, this haplotype information through the barcodes that are in the reads. And this was really, I think, a game changer for us for being able to study this chromosome. You'll see that in a moment why that is such a tricky thing to study. But in a nutshell, the methods we then developed to analyze these kind of data afterwards were basically to map everything from one perspective, for example, against the reference genome. So the zebrafinch has quite a very well-established reference genome. I will just refer to this here as the A chromosomes, the regular chromosomes, as a haploid assembly. So in soma, when we have 2 separate haplotypes for each chromosome because it's a diploid organism, we expect these same A chromosomes also to be in the germline, right? This is the shared genomic component, plus an extra haplotype if we have something that is very similar to the regular chromosomes, basically. That would be then from the GRC. So if we compare these 2 tissues, we can account for the variation on the A chromosomes. We can take that as a negative control, basically, and then we can find tissue-specific variation that we can then assign to this chromosome of interest, the GRC. And as it turns out, if some of these variants are inside of coding regions, protein coding regions, then we can also look for these on the RNA level, for example. So here, in this case, when we find transcripts that have these specific SNPs in here, we can say it's not just this gene of interest transcribed in this tissue, but it's that specific paralog on the GRC that's transcribed in this tissue. And in the same manner, we did this also on protein mass spectrometry data that we got through collaborations that were produced for a different purpose, but nicely also we're able to find specific, in this case, peptides where amino acid changing variants were present. So we can again then also say this specific paralog on the GRC is expressed and not the regular chromosomes, basically. So let's kind of walk you through some of the rather surprising results and kind of what this means for the next steps in this kind of research, because as I said, this is all like newly established methods for dealing with this kind of data types, and at the same time hoping that we can find a replacement for 10x Chromium. with different linked read methods in the future. So the first thing we tested was then to see, well, is that chromosome, like the W chromosomes I showed you before, just packed full with different repetitive elements? And short answer is no. And here's one way to visualize that. Here we have the so-called repeat landscape graph. This is basically on this axis the relative age of a transposable element, old transposons here, young transposons here. And on this axis, we basically have the difference between the proportion of repetitive elements in one tissue versus the other. And the idea being, if something was enriched on the GRC and you compare two different tissues from the same bird, then everything should be positive, basically. The graph should be going up above 0, basically, if that was the case. And as you can see, almost everything is kind of in the negative parts here. So what we think is actually the case is that this chromosome is not just not enriched, but it's actually, relatively spoken, impoverished for repetitive elements. And this, of course, left us very puzzled because we were kind of expecting somewhat similar patterns like with the W chromosomes, or at least kind of maybe something to compare against. But it turns out that still the GRC is repetitive, just in a different way than maybe one would expect naively, or as I was expecting naively. The idea being that we have high copy number regions that are very similar to the regular chromosomes, or extra paralogs, basically. So let me walk you through this way of visualizing that. The idea being, here we have now our regular chromosomes, the A chromosomes, so the autosomes, and then the sex chromosome here, Z only. We plot or we map all of our data against these different chromosomes, so from testes and soma from the same bird, and then we'd make a coverage ratio for each position or each window across these different chromosomes. And if everything was equal, if both genomes were identical, we would kind of expect a graph like this, right? Nothing would be different, basically, but instead what you see is there's just local peaks in coverage all over the place, basically, including something that was verified in the first molecular study on the GRC roughly 10 years ago. So that's already nice, consistent with previous data. But in this case, for example, we were able to say this region here, DPH6, is then the most copy number rich region. We expected to have 300 copies on the GRC, which we then can test using cytogenetics. So this is something that we then did using fish. So here we have a DAPI staining for DNA in blue, two nuclei from muscle, for example. In this case, there's no signal, because the idea being everything that's in single copy on the regular chromosome, so just diploid state basically, will not give a signal in the fish. But if you have this many, many hundreds of copies on the GRC, this is the signal that we then see here in the germline from the same bird. So really the idea being, this is a way to visualize and verify some of these high copy number genes. So to summarize that part, what we think this chromosome is composed of is regions that are a single copy on the regular chromosome, were copied over to the GRC and then expanded in copy number. So in a sense, they became repetitive, even though they're actually genes. So the emphasis really should be all the regions we looked at least the highest confidence genes that we have, 115 regions. These are all basically protein coding genes. And I'm just going to show you this gene ontology enrichment analysis just to show that this is not a random set of genes. I'm happy to speculate of what this could mean. I just want to say that strongest enrichment is in female gonad development and on also different early embryogenesis developmental terms. But I should also mention that we have around 200 additional genes that we're still verifying at this point. So this exact picture might change quite a bit, but it really looks like this chromosome is packed full with paralogs of a bunch of different genes across the genome. And in this case, basically, kind of like a tissue-specific copy number variation. I also mentioned before that we can then find these specific expression evidence or the specific SNPs on expression evidence, for example, in RNA sequencing and protein mass spectrometry. So this was the case for some genes in testes and some genes in ovary, maybe a few dozens actually. And I personally would say the strongest evidence, I think, for expression would be through the peptides that we found with amino acid changes. So 4 genes in testes and 4 genes in ovary. So no matter what this chromosome does, it does something that we suspect at least maybe has some potential function for the host, but that has, of course, yet to be elucidated, what that exactly would be. So I just want to leave you kind of with one last finding that we were rather surprised about, which is the idea that if we have expression evidence and we can find the sequences of this chromosome, can we then also maybe have an idea of how old some of these genes are on the GRC, right? If they were copied over, when did this happen? Just want to show you one phylogeny where hopefully this gets across the point. We have basically here a phylogeny of the achromosomal copies. This is all the birds we could also find in databases because zebrafinch here was the first germline genome that was sequenced. And down here we have the paralog now from the zebrafinch. The idea being, if these paralogs cluster far away from each other, the GRC gene was likely copied in the common ancestor of all these birds, these songbirds. And we were really like trying to refute this for a long time and like scratching our heads a lot about that. Is this just an artifact, etc.? But everything we tried, we always ended up with the same result, basically. And around this time when we were wrapping up the study, a very nice cytogenetic study came out that is now been published in PNAS, but was a bioarchive preprint back then. And they very nicely showed that every single songbird they looked at with their complementary methods, they did find a GRC. So I think these complementary lines of evidence taken together make me rather carefully optimistic that maybe we'll find actually the GRC in all songbirds where we look at those, basically. So just to kind of wrap up that part, I just want to kind of emphasize we could do this for a bunch of different genes, not just the old genes. And we left with something that kind of reminds me a bit of these sex chromosome strata that I mentioned earlier in this talk. We have some old genes, some intermediate genes, and some, or actually quite a bunch of very young genes. So we think this is actually a very rapidly evolving chromosome. And when we now trace back where these genes come from, because they're paralogs of regular chromosomal regions, it actually turns out that these come from all over the place. So here we have a placeholder for the GRC, and then each line connects them to where the paralog is sitting or where the donation has been happening from. You see different macrochromosomes, microchromosomes, even the sex chromosome. So this is a rather fascinating result, I think. But keeping in mind, the assembly is very fragmented, and this is what we're going to work on now. But I hope with that part, I could convince you that it's possible to study this. This is a repetitive chromosome, but not the repetitive things that we imagine would be on this chromosome. It is very likely present in different songbirds, maybe 5,000 species, if that applies to all of them, and rapidly evolving. And now, of course, we need to figure out what this does. Why has it been around for so long? If that's true, what does it do? Does it do anything of importance to the host? Is it just a very good selfish element? And we're trying to address these things by reconstructing evolution across songbirds for different species where we can get germline tissue sequence and also then developmental aspects of GRC elimination. And I just want to give you one last spoiler just so you kind of know what we're getting into now. And this is really big thanks to NGI Uppsala, who have been producing the first PacBio hi-fi data and Nanopore ultra-long read data for helping us to get a GRC reference for the zebrafinch. I just want to leave you with one first result that Francisco and the new postdoc in the group, Simon, have been finding now. This one region that I showed you before, DPH6, this most copy number rich region on the GRC, everything we assembled so far looks like it is very, very complex. It's basically a palindrome that is arranged in tandem after another. And each of these arms here is basically one copy of the gene, and this goes over and over and over and over again in many, many copies. So stay tuned for that. And with that, I really would just want to say big, big thanks to NGI and Scilab Lab and NBIS for all the help and collaborations over the last years, because really many of these things we wouldn't have been able to do the way this worked if it wouldn't have been for NGI to be early adopters of these technologies and also to work with us to test some of these things, both from the lab side as well as from the bioinformatics side of things. So big thanks to the Uppsala Genome Center, SNP and Seq, and also NGI Stockholm, and of course also NBIS long-term support. And with that, I just want to thank a lot of people. I mentioned the key people on the slides, and I want to thank the lab for being amazing in this pandemic and supporting each other. Funding also, of course, very important, and I'm very happy to take any question. Thanks so much.
Thank you very much, Alex, for a great talk. So it's now time for the Q&A session. So I think that all panelists can start sharing your videos. Yeah, great. So I think we can start with, I just want to say that you can still submit questions through the Q&A chat, and you can also, if you want to ask it in person, push the raise your hand button. So I think we can start with one of the questions that was sent in. So this question is for Remy. So can you elaborate on the distance distribution you observe for 10x linked read data and how it compares to the high C distance distribution you presented?
Right. So the thing with Chromium 10x and all linked reads essentially is that it's based, it uses based on DNA extraction, right? So as Olga mentioned, it's an art in of itself to get long fragments out from the extraction, and yeah, that's a huge problem. And also, as I showed you in the distribution of IC lengths, there were millions of sort of contact points. Each read pair is a contact point. But for linked reads, you operate with molecules, right? So each read pair is not a contact point, but comprised of several reads that are linked together. So that will also reduce the amount of the amount of longer contacts you will get from this method.
Thanks. So during the registration, we also had a couple of questions that was sent in beforehand. So I'm gonna read one of them. So one question that came in was that, is BioNano a good alternative to Hi-C? Who wants to answer that question? Maybe Olga?
Yes, they're not self-exclusive, they are complementary. So what you do in BioNano, you're doing the optical map that you can use for proofread assembly, you can use for phasing, you can use to elucidate the repetitive fragments and so on. But the thing is that with Hi-C, you're introducing extra level of base pair confirmation, because Hi-C is a way you sequence. So if you have some dubious indel that you cannot perhaps resolve in Nanopore or PacBio data, Illumina reads will give you extra level of confirmation. But when it comes to scaffolding and everything else, adding another technology is sometimes another sort of set to make an orthogonal confirmation of the things that you are looking at. Alex had.
Yeah, I can maybe also make a comment kind of related, of course, with a very bird biased perspective, but kind of what we've looked at before with comparing Hi-C and also optical maps for some other species that I didn't show today. I think optical maps are really nice for verifying some misassemblies and or spanning some gaps. But I think, at least for birds, they don't seem to be able to go along or across the centromeres. That kind of seems to be more like a biological issue. As far as I can tell now, bird centromeres might be like human centromeres, 1, 2, 3, 4, 5 megabases in length. And that's just beyond the molecule length that we get basically with the BioNano. And this is then the advantage of Hi-C, where you can have these contacts across one and another. So just kind of, I don't know if that applies to other organisms, but just from a bird perspective, maybe that's what we expect. We can very nicely get chromosome arms with BioNano, but yeah, that's maybe the limitation there.
Another question that I think a lot of people wonder about is, so what is the cost for reference genome? Maybe both Alex and Henrik can answer that.
The answer is it depends, because it depends on the ploidy level of the genome size. It also depends on the overall complexity of the genome, and also depends on the DNA. So the cost of your sequencing project, 90% would depend on the DNA quality. Because if DNA is good, we'll get, for the bird, for instance, it's enough with 1 or, in the worst case, 2 SMRT cells. But if your DNA is subpar, we'll need 3 or 4, doubling the cost. So it depends, but for the bird genome, I would say, Alex, you're better to answer. Around 150K Swedish?
Yeah, something like that.
Yeah, that's including the annotation and ultra long nanopore. Yeah.
I mean, the only thing I can add here is it depends a bit on like how you measure the cost also for curation, basically. So if you have somebody in your group to do that, and or collaborators, or through NBIS, for example, to help with that, I think now that, at least through the Vertebrate Genomes Project, etc., there's a bunch of different curation sets out there where basically if you have an organism that's not too far away from that, I think the curation time will be more predictable. And I think now for us, for example, also we're not going to spend the same amount of time as Valentina has done for this first paper and first genome. So we learned a lot from that, and also we know now what to expect, etc., or what to look out for. But if you're looking at an organism that is very far away from anything that's been tested in these ways, I would suggest significant time to allocate at least, or at least head scratching and blood, sweat, and tears for curating that as well.
Can I perhaps add there also when it comes to the bioinformatics cost, and I agree fully with what Alex is saying here. Genome assembly is a very explorative subject, I would say, and you can basically continue forever to polish and fiddle around with your assembly, and it's actually kind of hard to say, okay, now we stop, now we don't do anymore, because you can always do a little bit more. So you can spend an insane amount of hours there, and if you would ask, for example, NBIS to do this, we would be very happy to help. But the cost will increase the more time we spend on it. Annotation is less explorative, I would say. The manual creation of the genes would definitely leave to the user, but the cost in annotation would then also depend a lot on the organism and also what you in your group can do to help. For example, we want to use assembled transcripts in our annotation pipeline. If you yourself can assemble, the RNA-seq reads into transcripts, that's a saving. If we need to do it, there's a cost. So the cost for annotation could be anything from 30,000 to more than 100,000, depending on how much we need to do with the complexity of the organism and so on. Bacteria is basically pushing a button, almost.
Yeah. I mean, if you want to have like a good calculation of the cost, it's always a good idea to ask for a meeting. So we can, during the meeting, we can figure out what would be the recommended depth and so on and so forth. So meetings are free of charge and meetings are not binding. So that's the best way to find out the best setup. And then we'll also connect you with NBIS, and we can have a joint meeting together with NBIS to try sort of to estimate. But sometimes, as Henrik says, before you get the genome, you don't know how long time you will spend on annotating it.
So I can also mention perhaps that annotation can be a fairly time-consuming endeavor, because some of the analysis take a long time to run in the computer. You're not paying for this time, but don't expect to have an annotation, in a week. One run can be a week or 10 days or even more. And we need to run the tool several times.
Thanks. Yeah, it's a great point, Olga, that you can always set up a meeting with one of the project coordinators at NGI and also together with NBIS to clear this out. So we have 2 questions here for Olga from the Q&A chat. So the first one is, have you looked into using multiple displacement amplification to increase input DNA for PacBio sequencing, for example, for malaria samples?
Yes, it's a very good question. And yes, we did, and not just MDA, we tried different ways of whole genome amplification. So the problem is you're busting up the number of molecules that you can sequence, but you are severely reducing the complexity of the library. So also, if you want to sequence fragments that are long, the threshold lies somewhere around 5 kb, because if you go for the insert size that is over 5 kb, you'll be sequencing chimeric reads. So you will lose a lot of data. So we've done several projects using the MDA. The assemblies were not stellar, but as you say, in malaria, that's what you have to work with. Also, there is this ultra low input for PacBio that starts with a couple of tens or couple of dozens of nanograms of DNA. It does use NDA or MDA, but it's a little bit different. It's a biospecific protocol. It's still experimental, so I cannot give you, like, we're testing it right now. So contact me in a month or so, and most likely then I would be able to give you any hints. But there are ways around it, but MDA is not the best way to sequence genomes. However, if that's the only option, then it's the only option, just understand the limitations. And then the second question also I've got, yes, have you looked into long read sequencing single cell RNA? Yes, we did. We've done it mainly on humans, but We're also testing on other things. And I think on bioRxiv, there is a recent paper by my colleague Adam Ahmed and with other collaborators in Stockholm from single cell facility. So we sequenced the entire human genome from a single cell. So it's still experimental, but we're working on it and we're collaborating with both NGI nodes on that.
Great. So another questions that was sent in beforehand is, so I know that it's preferable to use PacBio and high-C assembly on the same individual. However, how bad is it if I don't?
Can I perhaps answer something there?
Yep.
So, I mean, that totally depends on the organism. We've been working on, for example, barnacles, marine organisms, where you have two things sitting next to each other in the same aquarium, but you can't align the reads from the one individual to the next one. But if you have some other, much less variation when it comes to comparing individuals, it could be fine. So it totally depends on the organism and the material you can access, I would say. Mm-hmm. You might even be working with, like clonal organisms almost, that are very close to each other.
Mm-hmm. I can just pitch in there. So one of the examples where we could use different individuals, that was herring. The heterozygosity level is relatively low. So it all depends on the heterozygosity level. As Henrik said, like barnacles, they are on the extreme range, and there you cannot. But yeah.
Great. So another question, I think for Remy maybe. So how long will the scaffolding take once you have sequenced the libraries?
Sure. So for just running the scaffolder itself, it shouldn't take longer than a week, right? So, I mean, there are some very large and complex genomes, and there, for instance, 3D DNA, in my experience, it will not work at all. So, I mean, it can run for a month and you still don't get any results, right? But SALSA 2, it shouldn't take longer than a week or maybe 10 days. Yeah.
Okay. Another person here who has both old PacBio data and new HiFi data. So is it possible to mix these types of data?
Unfortunately, there is no good way to do it because the modern PacBio assemblers, they are using HiFi algorithms, or they really need this QV20 and up to get a good result. So unfortunately, not really, but you can use HiFi data as a, you can make assembly based on that. And then you could map your old PacBio data on top of the new assembly, but you cannot mix them in the same assembly from scratch, no. No.
Okay. Another question here is how deep should one do the sequencing?
Depends on heterozygosity of the organism. We usually say 15x per allele minimum, but we also have seen that the more data you put in, the better quality of the assemblies you are getting. So, but after a while, you start saturating you start to saturate your library. So you sequence every single molecule that has come from the same library. So if you want to have super duper genome, perhaps you could sequence a couple of libraries at 15x. But yes, 15x for PacBio per allele. On Nanopore, at least 20x per allele, rather 30. And then Remy perhaps could say how much we need for high-C.
Yes, so the rule there is about 200 million reads per gigabase of genome. I mean, you can get away with less and more, right? But that's just the rule.
Mm-hmm.
Alex, you wanted to add something there?
Also, just one thing to keep in mind here is that it depends a bit on what you sequence. So if you sequence, for example, heterogrammatic sex, like a female bird, then if you want to look at the sex chromosomes, you will always have half the coverage relative to the rest. So I would, by default, always say try to go for twice the amount of coverage as you would go for a male of that bird, for example, or for mammals, and of course the other way around, for more coverage for males than females. Then just to make sure that you get this Z and W or the X and Y nicely covered there.
So another question I have here is, so my tissue sample was not obtained or stored in an optimal way. So can it still be used for Hi-C or for PacBio analysis?
I can answer PacBio. So we have an article in review now in GigaScience when we compare different tissue preservation methods and DNA extractions. So the good thing about, so most likely the answer is for Hi-Fi, most likely it will be fine, but you might not be able to get ultra long molecules for the nanopore. Yes, so we need to extract and see. If it's stored in -20, we got sometimes extremely good Hi-Fi libraries, and in some cases they were dreadful. So it's again, it depends on the sample. Blood usually does much better than muscle. Muscle is, in our experience, is one of the worst tissues to work with. Too little DNA, much more degraded. And Remy for Hi-C? Yeah, Alex also.
Yes, I can just say for Hi-C, I mean, I try to stress that suboptimal storage is it's a big no. So we will try to discourage that, but of course, sometimes it's precious material and you can't really do anything about it. But I mean, if it's stored, for instance, in RNA later or ethanol, we might try it, but you can, or yeah, we might not try it, but you can at least, the manufacturer says that you might try it, but we won't, right?
A note here, that's not published data yet, but RNA later and Hi-C is not a good combo. You're losing lots of cis transcript or cis orientations, so no. Spoiler.
I can maybe comment on both of these points. Maybe I should have made this clearer, but for this paradise crow that I showed today, this was maybe a prime example of a suboptimal sample, basically, because this is a bird that lives only in Indonesia, and you can't really you can't take like liquid nitrogen with you, etc. So this was actually one sample was in DMSO and then fridged for a long time or room temperature for a bit, etc., and then transported to Europe, and then the other sample was RNA later, which we use for the Hi-C. So it does work sometimes, but it does, of course, no guarantee that it will work for your sample. I just wanted to kind of mention it can work if you're willing to take that risk. I guess in our case, we just didn't have any other samples, so we had to try that. But it did not work, for example, for optical mapping or nanopore. So that was just too fragmented for that. It was okay-ish for the 10x, could have been better. Was, I think, good enough for PacBio and also worked okay enough for the Hi-C. So with that in mind, like keep that in mind. What is the things you definitely need to get from your sample, and then see what's the risk you're willing to take, maybe.
To add to that, so very often people ask, so what is the optimal? Absolutely optimal is that you collect and dissect in the field in the dry ice or directly snap freeze it in liquid nitrogen without a buffer. But we know that if you're going to a different country or remote location, liquid nitrogen and dry ice is not an option. Absolute ethanol will in most cases do just as well. After ethanol goes DMSO. But no DNA guard, no RNA guard, that is not suitable. And as soon as you come back to your lab, place the sample in -70 or -80. -20 is okay for a couple of weeks, but long-term storage, it should be done in -80.
And Olga there, so once you've collected the sample, you know, you should take care of it very, very quickly, right? If I'm thinking about RNA, for example, that is quickly degraded. So if you, for example, need to collect and kill an animal, you should try to preserve it as quickly as possible after death.
Yes, or you can split the sample in two. For RNA, put an RNA later, that will work. But for all the DNA work and the chromatin and so on, ethanol or something like that.
So let's see, I think we have one more question here in the Q&A chat. So how much improvement in scaffolding do we expect from Omni-C method compared to Hi-C for a typical genome?
I don't think we have done like direct comparisons. I mean, the thing about Omni-C is that you get a more complex library, so you can get away with fewer reads, right? But the information is in theory the same, right? But with other complexity. So I don't think it's very easy to answer that one.
Thank you. So I don't think we have any more questions. So if you do, you are welcome to contact any of the project coordinators again at NGI and also NBIS if you have more questions. So I will end this webinar by sharing my last slide. So again, thank you all for attending today's NGI and NBIS webinar on the novo reference genome sequencing and analysis. And I would like to take this opportunity to really thank all our fantastic speakers today. And we all hope to see you again very soon at any of our upcoming events during the fall. So we already have one of them planned, which is more details on the Oxford Nanopore system that we provide here at NGI. And please provide us with feedback on today's session, and in particular on what you would like to hear or know more about from us at NGI and NBIS. So with that, I will end this webinar and yeah, bye-bye and have a great day, all of you.
Bye.